Genes provide the instructions for making specific proteins, but a gene does not build a protein directly. The bridge between DNA and protein synthesis is the nucleic acid RNA. However, remember that nucleic acids and proteins are written in two different chemical languages. One in nucleotides and the other one in amino acids. Getting from DNA to RNA and to protein is governed by a molecular chain of command with a directional flow of genetic information, a concept dubbed as central dogma by Crick. Transcription is the synthesis of RNA using information in the DNA. Just as a DNA strand provides a template for making a new complementary strand during DNA replication, it also can serve as a template for assembling a complementary sequence of RNA nucleotides. For a protein coding gene, the resulting RNA molecule is a faithful transcript of the gene's protein building instructions. This type of RNA is called messenger RNA or mRNA because it carries a genetic message from the DNA to the protein synthesizing machinery called ribosomes. Remember that DNA is a double-stranded molecule, and RNA or mRNA on this matter is a single-stranded molecule. Thus, only one strand of the DNA is used as a template in producing mRNA. This strand is called the template strand, which is the 3' to 5' DNA strand. The 5' to 3' strand, on the other hand, is called the non-template strand. The produced RNA therefore runs from 5' to 3' and is actually identical to the 5' to 3' DNA non-template strand with thymine replaced with uracil as previously explained. So for example, we have 5' TGG CTCA 3', which is the non-template strand, and 3' ACCAAA CCGAGT 5', which is the template strand. Our mRNA molecule sequence would be 5' UGGUUGGCUCA 3'. The length of DNA that is transcribed into an RNA molecule is called a transcription unit. Each transcription unit has a sequence just above the strand, or as biologists call it, upstream to the strand, and that sequence is where the transcription unit is going to start. This sequence is called the promoter, and it almost always contains a sequence of two of the nitrogenous bases, specifically a repetition of 5' TATAAAA 3', and on the complementary strand 3' ATATTT 5'. This is called the Tata box and is nearly universal and helps transcription enzymes and factors to figure out where to bind to the DNA strand. Upon these transcription enzymes and factors, collectively known as transcription initiation complex, bound to the promoter sequence, the RNA polymerase moves along the DNA to unwind it and starts transcribing the 3' to 5' template strand. Just like the DNA polymerase, RNA polymerases can assemble polynucleotide only in the 5' to 3' direction. Unlike DNA polymerases, however, RNA polymerases are able to start a chain from scratch. They don't need a primer. As the RNA polymerase moves, it rezips the DNA behind it and lets the growing strand of mRNA peel away. Eventually, the RNA polymerase reaches another sequence downstream called termination signal that triggers it to pull off. Now, before the mRNA leaves the nucleus to initiate protein synthesis in the cytoplasm, some modifications are done through a process called RNA processing. First, a special type of guanine is added to the 5' end and is called the 5' cap. This process is complicated, but two key players are involved. The first one are small nuclear ribonucleoproteins, abbreviated as NURBs, which are a combination of RNA and proteins that are able to recognize the sequences that signal the start and the end of the areas to be spliced. SNRPs bunched together with additional other proteins form spliceosome, which what does the actual editing process. The protein coding sequences of the mRNA that remain are called the exons because they will eventually be expressed, and those that are removed because they are non-coding are called introns, which will stay in the nucleus and get recycled. After all those RNA processing, the processed mRNA is now free to leave the nucleus and be translated into proteins by the ribosomes in the cytoplasm.